أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم إن تنصروا الله ينصركم ويثبت أقدامكم صدق الله العلي العظيم. Today I will speak about prosthetic groups, cofactors, and coenzymes. Many enzymes contain small non-protein components that participate directly in substrate binding or in catalysis. Termed prosthetic groups, cofactors, and coenzymes, they extend the repertoire of catalytic capabilities beyond those afforded by the limited number of functional groups present on the amino acid side chains of the peptides. Some enzymes require no chemical groups for their activity other than their amino acid residues. Others require an additional chemical component called a cofactor, either one or more inorganic ions such as iron 2+, magnesium 2+, or zinc 2+, or a complex organic or metalloorganic molecule called a coenzyme. Most are derived from vitamins, organic nutrients required in small amounts in the diet. Some enzymes require both a coenzyme and one or more metal ions for their activity. A coenzyme or metal ion that is very tightly or even covalently bound to enzyme protein is called a prosthetic group. Actually, the prosthetic groups are tightly and stably incorporated into the protein's structure by covalent or non-covalent bonds. For example, cytochromes have an iron porphyrin complex as their prosthetic group. Metal ions constitute the most common type of prosthetic group. Roughly 1/3 of all enzymes that contain tightly bound iron, copper, cobalt, magnesium, manganese, and zinc are termed metalloenzymes. Metal ions that participate in redox reactions are generally complexed to prosthetic groups such as heme or iron sulfur clusters. Metal ions may also facilitate the binding and orientation of substrates, the formation of covalent bonds with reaction intermediates, for example, cobalt 2+ in coenzyme B12, or by acting as Lewis acids or bases to render substrates more electrophilic or nucleophilic and hence more reactive. A complete catalytically active enzyme together with its bond coenzyme and or metal ions is called a holoenzyme. The protein part of such an enzyme is called apoenzyme or apoprotein. Coenzymes become bonded during the enzymatic reactions. When joined with a coenzyme, the inactive form of enzymes, also called apoenzymes, form active holoenzymes. For example, dehydrogenases utilize either nicotinamide adenine dinucleotide plus or NAD plus, or nicotinamide adenine dinucleotide phosphate plus or NADP plus. These are among the most important coenzymes. The function of these coenzymes is to accept 2 electrons and 1 hydrogen ion from substrate, thus oxidizing it. The 2 electrons of NADH can then be transferred to a second molecule which will become reduced, namely it gains electrons. In the cells, the energy producing catabolic enzymes use NAD as coenzyme. The synthetic processes, however, use NADPH as hydrogen donor. In many coenzymes, as in NAD plus and NADP plus, which contain nicotinamide, the essential components are vitamins, particularly those of B group. Some examples are pantothenic acid or vitamin B5, which forms part of the important coenzyme A, riboflavin or vitamin B2, incorporated into the molecules of flavin adenine dinucleotide or FAD, pyridoxal or vitamin B6, a coenzyme of transaminases and decarboxylases. At the end of this class, I draw your attention to the summary of what I said in this class. Cofactors are either essential ions or coenzymes. Essential ions are either loosely bound or tightly bound to enzymes. Those that are loosely bound to enzymes are called activator ions, and those that are tightly bound to enzymes are metal ions of metalloenzymes. Coenzymes also are either loosely bound or tightly bound to enzymes. Those that are loosely bound to enzymes are called co-substrates, and those that are tightly bound to enzymes are called prosthetic groups. Please pay attention that metal ions constitute the most common type of prosthetic group. والسلام على من اتبع الهدى.